Stem cells are characterized by the ability to renew themselves through proliferation with the potential to differentiate into a broad range of specialized cell types. These cell types can be found in all multicellular organisms. Mesenchymal stem cells have been shown to be a valid source for at least, but not exclusively, 3 cell populations: adipocytes or fat cells, osteoblasts or bone-forming cells, and chondrocytes or cartilage-forming cells. Major sources for mesenchymal stem cells are adipose tissue, bone marrow, placenta, umbilical cord blood. Ultimately, mesenchymal stem cells provide an accessible tool available in all adult organisms to treat injuries by regeneration of the tissue and structural restoration. Fat stem isolates and stores stem cells from the fat tissue of different animals and differentiates them into cell types that can be used for regenerative therapies in close collaboration with leading veterinary clinics in Europe. Companies such as FatStem in Europe have developed procedures that are easily utilized by veterinarians and which are becoming increasingly popular as news of the success of such a technique continues to spread. FatStem's product development facility has designed an elegant all-in-one kit providing all necessary recipients and documents required for adipose tissue biopsy and blood collection. This kit is the result of practical product design and refinement through veterinarian feedback. The kit is sent to the vet by the FatStem courier or provided upon product presentation on site. With the kit at his or her disposal, the veterinarian plans a fat biopsy from the patient in the clinic. Adipose tissue is taken at the tail of the horse, under local anesthesia, and under aseptic conditions. FatStem researchers have shown that adipose tissue-derived stem cells are a very suitable source for harvesting stem cells from adult tissues. This tissue is easily accessible in humans and animals using minor surgery, liposuction or fat resection. After disinfection, an incision is made to resect 20 to 30 grams of fat tissue, which is transferred into a sterile tube filled with a physiological solution. Finally, the wound is sutured and disinfected. Compared to bone marrow, this procedure holds fewer risks to collect and no comorbidities have been reported up till now. Moreover, this tissue allows for the highest yield of mesenchymal stem cells per tissue weight in comparison with other sources. The rich mixture of cells that can be freshly obtained from adipose tissue contributes to the optimal growth of mesenchymal stem cells. During the same procedure, blood is collected from the horse. The addition of autologous serum and platelet-rich plasma, or PRP, to these cells stimulates their proliferation potential even more. PRP and serum are also derived from blood drawn from the equine patient. These blood products contain high concentrations of growth factors that have the potential to boost mesenchymal stem cell proliferation. In this way, the large initial amount of mesenchymal stem cells combined with an optimal growth environment results in a jumpstart cell culture in vitro, quickly giving rise to in vivo usable cell numbers for stem cell therapy. All recipients are placed again in the styrofoam box for transportation, separated by the gel pack supplied in the kit. The barcode labels provided in the kit are attached to the biopsy tube, blood bag or tubes, and patient identification form. The vet fills out the identification informed consent form and completes the package for its return to FatStem Laboratories. FatStem is notified about the biopsy by the vet, and the company arranges a pickup of the kit in the clinic. Upon arrival in the lab, FatStem immediately starts the isolation procedure. The cells are released from the adipose tissue by enzymatic digestion. This treatment processes the tissue to a single cell suspension, which will allow an optimal attachment of the cells to the plastic recipient. These attached cells can then proliferate and further grow on the cell culture flask surface. Cells in vivo and in vitro require growth factors for their growth and proliferation. For this purpose, the blood collected by the vet is centrifuged in the lab to separate serum or plasma from the blood cells. In a complete medium, the cells from then on grow in a bioincubator till the cultures are confluent, or in other words, when the cells will be harvested. At several time points, the cells are counted to monitor their growth and cell viability is determined. To check their stem cell characteristics, researchers at FATSTEM laboratories screen for their ability to present several specific markers. Cell staining methods are used to pinpoint and assure their stemness in the lab. Finally, the FATSTEM experts screen the potential of the cells to differentiate at least in fat, bone tissue and cartilage. All quality data is stored at FATSTEM's databases. Only when they pass these quality controls, the cells are certified and can be processed for delivery. FATSTEM certificate is always added to the stem cell product, and together with the barcode label, accurately allows traceability of the stem cells. For delivery, usually after 10 to 14 days, all stem cells can be harvested from the culture flasks, counted, and for the last time their viability is assessed. The cells are then prepared as an injectable cell suspension. This cell suspension is now ready for shipping in certified tubes back to the veterinary clinic. This cell suspension is ready to inject for the vet. Stem cell applications in veterinary domains are mainly on horse orthopedics, from tendons to ligaments and joints, as well as in dog osteoarthritis. A diagnosis of lameness is never the news a horse owner wants to hear, but it may be reassuring to know about the innovative and effective therapies now available at different clinics working together with FATSTEM laboratories. Lameness can be caused by the degeneration of a joint, arthritis, or it can result from a tendon or ligament being acutely overloaded or injured. After a tendon injury occurs, mature cells invade the area and produce scar tissue. This is a slow process and the repaired tissue is inferior in mechanical properties as compared to the original tissue, predisposing to poor running efficiency, recurrence and repeated injury in up to 80% of affected horses. They account for nearly a third of all equine injuries that occur during racing, with a reported prevalence of up to 50% of the horses. Obviously, once a horse has damaged a tendon or ligament, the risk of re-injury is very high. Stem cells can lower this risk dramatically. Fat stem cells have the potential to reverse this damage and restore the tissue's normal structure instead of just repairing it, usually leaving a weak spot, often prone to new injuries or re-injury. These stem cells have the ability to soundly integrate into the tissue and form a new, solid structure. Applied at the site of injury, fat stem cells can differentiate and replace damaged cartilage of joints, form new tendon or ligament tissue, and even glue hydroxyapatite crystals with collagen fibers and other proteins to regenerate bones. This therapy shows to be very promising, as up to 80% less re-injuries can be achieved. After rehabilitation, these horses can perform again at the highest sport level. Because of the quick delivery of the stem cell samples, already after 10 to 14 days, injuries can be treated quickly, thereby increasing regeneration before weakening scarring appears. FATSTEM developed a new product, PlateStem, which is considered a combinatory treatment of stem cells and growth factors, resulting in an even quicker healing process. This stem cell treatment also uses autologous mesenchymal stem cells from the horse's adipose tissue, but this sample is supplemented with platelet-rich plasma, or PRP, from the same patient. This treatment helps assure that the horse's injury is healed, but the risk of re-injury is still higher compared to stem cell treatment. That's why FATSTEM Laboratories made a combination therapy with stem cells, PlateStem. When plate stem is injected into damaged tendon, stem cells are stimulated by the growth factors to reproduce new cells at the injured site. Surgery, platelet-rich plasma, and stem cell treatments all need to be used with careful rest and rehabilitation. A controlled increase in exercise is still an important part of the therapies. The goal is to return the injured tissue to a more resilient and more normal condition than what could happen without these treatments. Often horses and their owners are confronted with serious injuries of the joint cartilage. Because of the large joint volume and the pressures arising from high performance levels, FATSTEM Research Facility has found stem cells alone to be unsatisfying for full recovery and regeneration of the cartilage. For this purpose, FATSTEM has developed a new product specifically designed to meet this challenge, named ChondroStem. The gel is specifically suited for intra-articular pressures, keeps the mesenchymal stem cells in place, and guides them to the injury site. This product is capable of regenerating cartilage tissue, and horses can fully recover in this way to again attain the highest sport levels. FATSTEM, as an animal healthcare company, will soon launch cutting-edge technology with a collection of equine umbilical cord blood stem cells from the afterbirth of newborn thoroughbred foals. The process will be named CordStem. The collection and storage of the adult stem cells from the umbilical cord in readiness for stem cell therapy is marvelous technology that will help reduce equine wastage. The technology involves the collection, growth, and cryopreservation of adult stem cells isolated from the umbilical cord. The stem cells will be stored at the FATSTEM storage facility and can be used later to repair injured tendons and ligaments in the horse from which they were extracted. Stored cells can also be used for future therapies as they emerge.